Hello, welcome to the final video of Chapter 2. This is about ATP, nucleotides, and nucleic acids. In this video, we're going to be talking about the structure and function of ATP. We're then going to be talking about other nucleotides that are around in addition to ATP, and then we're going to talk about the structure of nucleic acids. So nucleotides always have the same three components. Let's take a step back. The nucleotide is the monomer of a nucleic acid. Nucleic acid is a polymer made up of monomers, which are nucleotides. Okay, so monomer, nucleotide, polymer, nucleic acid. So our nucleotides are always made of the same three pieces. First, we're going to have a nitrogenous base, and nitrogenous just means it contains nitrogen. In the case of ATP, our nitrogenous base is adenine. Then we're going to have a sugar, and the sugar is a monosaccharide, so it's just a one-ring sugar. And our sugar in ATP is ribose. Notice the OSE, that gives it away that it's the sugar. And then all nucleotides have one or more phosphate group. ATP has 3 phosphate groups. So here's the structure of ATP. We have our adenine, then our 5-sided ribose sugar, and then 3 phosphate groups. 1, 2, 3. So ATP is the body's energy currency. This is how we store energy, transfer energy, make reactions happen. The energy ATP has comes from exergonic reactions, and exergonic reactions are any reactions that release heat or light as the reaction is happening. This can happen very, very fast when the body needs energy to do some sort of physiological process. And what happens is this last phosphate group is released. Okay, so let's talk about this for just 1 second. Do you guys notice all these negative charges that we have going on here? We have this whole series of negative charges. Imagine for a minute how difficult it is to hold the negative end of 2 magnets together. They really don't want to go together, right? Imagine putting 4 of them all together. They repel each other very, very strongly. And so as soon as this last bond between the second and the third phosphate groups is released, this phosphate goes flying off, releases a ton of energy, and your body can then harness that energy to do something that it needs to do. Okay, so this is about several negative charges being all crammed together in a very close space, and how much energy we release when we finally let some of those charges go. When that bond between the second and the third phosphate group is broken, that happens by an enzyme called adenosine trisephosphatase. Okay, notice the ASE. ASE tells us it's an enzyme. So we abbreviate this ATPases. Okay, the ATPase is the enzyme that lets go of that bond. And that releases that, we call it an inorganic phosphate. We abbreviate that PI. And a PI is the phosphate with the 3 oxygens and with 4 oxygens, 3 of those oxygens having a negative charge. So here's my P, here's my 4 total oxygens, and here's 3 of those oxygens having a negative charge. Okay. This makes all these PIs, inorganic phosphates, floating around out there. We can bring those phosphates into different molecules and have them be functional groups that do really useful things in so many cases. We have a whole nother group of enzymes that adds phosphate groups to different molecules. We call those kinases. So let's talk for just a second about how ATP is produced. Okay, so we have glucose, our good old glucose 6-sided sugar coming in. It goes through the process of glycolysis. And then, under normal conditions, this leaves us with 2 3-carbon pyruvic acids. Those go into the Krebs cycle. You may have also heard this called the citric acid cycle. The Krebs cycle spits out a little bit of ATP. Glycolysis spits out a little ATP too. Krebs cycle spits out a little bit of ATP, but the big product of the Krebs cycle is high energy electrons that get carried to something called the electron transport chain. And the electron transport chain is a series of reactions that are decreasing the electron's energy and harnessing that energy to produce ATP. Now, that was the 30-second, very simplistic version of the story. I can tell you that if you take a biochemistry class, the entire class will be about how this process works in detail. There are 10 steps here. There are, I believe, 10 steps here. This is a whole nother series of steps. This picture is as simplified as you can possibly get. This is actually incredibly complex. The other nucleic acids that we are super interested in are RNA and DNA, and I'm sure you guys have heard about these at some points or another. But let's talk through them real quick. RNA is a single strand, while DNA is a double strand. Okay, in RNA, we have uracil, and it matches up with adenine, and cytosine matches up with guanine. We actually have 3 different types of RNA. Okay, there's messenger RNA, and that's what we normally picture when we think of RNA. And this carries information from the DNA in the nucleus to the ribosomes in the cytosol, where proteins get made. Our ribosomes themselves are actually made of a big chunk of RNA. So we have rRNA, or ribosomal RNA. And we also have small bits of RNA called tRNA, or transfer RNA, that haul around the amino acids that are getting added to the protein. And these two we're going to spend quite a bit of time talking about later on. So RNA, its whole job is to carry the genetic message from the DNA in the nucleus out to the ribosomes in the cytosol where proteins get made, and that allows us to produce our amino acids. DNA, on the other hand, is double-stranded. We know that thymine pairs with adenine, cytosine pairs with guanine, A goes with T, C goes with G. If you have a mismatch there, it can lead to all sorts of DNA damage, and if it's not fixed, possibly cancerous conditions. DNA typically stays in the cell nucleus. The DNA is incredibly long. The DNA is made of millions of nucleotides. And that's because the DNA is the complete set of instructions for genes. And all genes are instructions for making proteins. So DNA is the whole recipe book, the whole cookbook for making all the proteins your body could ever possibly make. So that's why DNA is so big compared to RNA. And we can see a little bit of a visual here. We have single strand RNA, double strand DNA. We have uracil in the RNA where we don't have uracil in the DNA. Both have the sugar phosphate backbone, and then they both have the base pairs.